Welcome to Mathematical and Computational Modeling of Cancer seminar. I'm very happy that our speaker today is Kit Curtius from UCSD. She's an assistant professor in the Division of Medical Bioinformatics at University of California, San Diego. She obtained her PhD in applied math at the University of Washington and did her postdoc at the Barts Cancer Institute in London. She was recently awarded the Leah Edelstein Cashett Prize from the Society of Mathematical Biology, and I'm very happy that she is speaking in our seminar. Please let me know in the chat if something is not working, and yeah, we can start.
Great. Thank you so much for the invitation, Nirvana. It's nice to be in some of these inaugural sessions. So yeah, my name's Kit, and I'm going to tell you a little bit about projects in the lab, some published, some pre-printed, and some preliminary results of unpublished work. And yeah, if you can't see anything, please put in the chat. So the focus of my group has been around cancer early detection and prevention. And I like this example of colorectal cancer to show why this is so important, where the 5-year survival rate of colon cancer caught at a local stage is over 90%, while colon cancer caught at an advanced distant stage has less than a 10% 5-year survival rate. Unfortunately, those diagnosed with CRC, only 39% of them are diagnosed at that local stage. So the goal is really to diagnose more of these individuals at an early stage when their cancer is more treatable. And two kind of words that come up a lot is prevention. And in colon cancer, we know that there is a precursor lesion called a polyp or an adenoma, where if you remove that precursor lesion before it becomes cancer, it's very effective in preventing cancer and cancer mortality. And then early detection would mean catching them at these very early stages when cancer has just started growing. And this is all kind of clinical information, but we know, and from previous lectures, we've talked about how cancer on a cellular level is actually an evolutionary process. So we have decades of omics research now showing us that this is true, where cells are undergoing selective pressures in certain ecosystems in microenvironments of the body and certain organs, and maybe certain mutations cause a selective advantage of a certain clone that may be able to outcompete its neighbors and grow into a larger population that will potentially malignantly transform and further might go on to form some sort of metastasis in a distant site. This is all based on processes that were described by Charles Darwin, things like genetic mutation, genetic drift, and natural selection. So this is the perspective that we take. And the ultimate goal, I think, of screening in cancer as prevention and early detection. It's identifying those patients at highest risk of developing life-threatening cancer and intervene to reduce or eliminate that risk. And so for various cancer types, we know that cancer screening is very effective, as I mentioned, and there's US guidelines and other nation guidelines as well for certain cancer types, for example, colon, breast, cervical, and lung, to start cancer screening at a certain age in certain high-risk populations, apply the test, see the result, and decide whether the patient can go on living their lives and not worry about cancer for a while, or potentially have some sort of intervention. And so lots of questions that we focus on in my group are, how is these guidelines decided currently? Are they working very well? And how many lives could we save possibly with improved screening strategies? And how those guidelines are made now are pretty basic and sort of heuristic. So current guidelines are often based on sort of limited clinical data and rely a lot on expert clinical opinion. And so, for example, a disease that we'll talk about today is inflammatory bowel disease, or IBD. And in patients with IBD, they have a higher risk of developing colon cancer. And so 8 years after they're diagnosed, they're suggested to have a screening colonoscopy to look for precursor lesions. And then after those findings, they're determined to have certain screening intervals of 1 to 3 years based on if any sort of precursor lesions are found for the rest of their lives. And so these are very kind of one-size-fits-all strategies that aren't tailored too much to very much information besides a few clinical variables. And so one question you could ask is, who decided this would be 8 years that the next screening colonoscopy should be done? And this is actually based on very, very limited data. Just basically, there's one Kaplan-Meier plot published in Gastro in 2006. They looked at the time to progression to some sort of neoplasia or cancer, and they saw that the probability of getting cancer goes up, or the becoming cancer-free goes down around 8 years. And that was kind of all the data that was used to decide that interval. So not a lot of computational modeling or understanding of carcinogenesis to inform these strategies. But again, as we know, the underlying cellular dynamics we know is what drives cancer progression. So the two Precancers that we'll talk about today a lot in the talk is Barrett's esophagus, which is the precursor to esophageal adenocarcinoma, and inflammatory bowel disease, which is a precursor to colitis-associated colorectal cancer. And so there's various studies now that show that actually these clonal diversity and the growth of these clones is actually a good biomarker for future cancer progression. So in Barrett's, we know that things like numbers of clonal populations predict cancer in Barrett's esophagus. And in IBD as well, we know that the large clones produce more cancer risk just by nature of having more opportunity for malignant transformation. So in my view, these computational models and mathematical models are needed to answer some of these complex questions that go up from epidemiological and clinical data all the way down to the cellular level to answer things about costs and benefits. And they're able to incorporate multiscale data. So again, epi data, clinical data, and genetic data, maybe germline or somatic, which we'll talk about. So my group, we call a Quantitative Cancer Control Lab. We try to answer some of these questions. We work really closely with clinicians to try and keep our modeling as relevant and as translational as possible. So things like, when should we recommend screening? How should biomarkers be utilized? And what are the costs and benefits of these strategies? So my PhD student, Brian, and I wrote a perspective piece in Nature Gastro this year or last year, kind of describing the idea of digital twins in oncology. And so you can think of digital twins as kind of these computational models that can look at outcomes in certain in silico strategies. So things like natural history of colon cancer, where you have 1 million people in a population and microsimulate what happens, and compare that in silico with what would happen with a certain screening strategy, and compare the kind of lives saved or other types of cost benefit analyses to determine best screening intervals. And these are actually the same type of models that were used in the most recent USPSTF guidelines on screening the average risk population in the US for colon cancer at age 45. So very important kind of modeling in the population health space. Then you could also think of things on the patient level. So when a patient comes in, they have multiple data types accumulated from the doctor. So things like imaging, clinical variables, genetics, histology. And ideally, you'd have some model that would tell you what's your risk of progression to cancer, say, in the next 3 years, and when should your next follow-up exam be undertaken. And maybe a model can help you decide that. And maybe the model eventually says, actually, the risk is so high now that you should intervene. So that's sort of on the patient level. And I think there's so much opportunity in this space where we're trying to work in our group. And some of the open questions are really, what's the data that's needed for your exact question that you're trying to answer? And what sort of models are appropriate? Do you need to model the entire human body? Do you need to just model one organ, et cetera? And what's really needed to get this clinically implemented? And so in our group, we study a lot of gastrointestinal cancers at the moment. And something that always kind of is interesting to me is that there's a lot of similarity across gastrointestinal cancer evolution, especially in the precancer stages. So we'll be talking about Barrett's esophagus today that has these stages of dysplasia and cancer development. Similarly, in the stomach, Helicobacter pylori infection often causes gastric intestinal metaplasia, which has the same sort of sequence. And again, IBD in the colon also goes through these stages. And interestingly, what we know from clinical data is that the progression annually from, say, Barrett's, GIM, and IBD to cancer is about 0.3% per year. So it's a really low progression rate and very similar, actually, across these cancer types. And one of the reasons for that is that the evolutionary process of this cancer is very similar across these organs. So all of these tissues have a crypt-like structure that's like the intestine. And what can happen is driver mutations can occur in, say, tumor suppressor genes in a stem cell, which then can clonally convert the crypt, which might give it a selective advantage, and it can undergo crypt fission and cause these clonal patches to form with, say, copy number alterations that might be driving what we call field cancerization. And this is just data from my previous group in London looking at this in CCO deficient patches of clones in the colon, where you can see sort of an on-phos view of the crypt here with these patches shown in blue, or sort of an on-phos view of the crypts growing. So this is just sort of the basis of a lot of the projects in the group is how these clones grow in the body. So the outline for my talk, I'll give a little background about these carcinogenesis models, and then focus on projects in Barrett's esophagus and esophageal adenocarcinoma and IBD and colitis-associated colorectal cancer. Okay, so again, normal cells in our body are undergoing these processes of evolution all throughout our lives. They face these forces of mutation, natural selection, and mathematical models of this process allows us to describe this pattern and pace of evolution toward cancer. And one of the models that we focus on in our group has been developed since the 1950s, which are these multi-type branching process models capturing these stages of progression from normal to cancer. And so again, with the colon cancer example, we know now with genomics that initiation of adenomas happen quite frequently in the average population, but they might go extinct because they're outcompeted by other clones. But some of them will take actually decades to develop over time to a large enough size that maybe one cell also malignantly transforms. And that would be sort of a preclinical cancer, and then a certain amount of time until symptoms develop. And those can all be, processes can be described by these stochastic processes. So things like inactivation of APC with 2 hits of that driver gene can initiate an adenoma, and these multi-type branching processes can follow these clone sizes and number, wherein adenomas can also malignantly transform with some rate and have some birth-death process that describes their growth, and then some eventual detection rate. And the nice thing about these models is that, you know, for over 75 years now, they've been connected to some of the best data that we have in cancer, which is high quality epi data, and often looking at cancer incidence rates. So the hazard function, or the incidence rate of cancer, is the number of new cases in a specified period divided by the number of person years at risk. And so some of that data shown over here on the right for colon cancer from a database called Surveillance Epidemiology and End Results Program in the US shows this age-specific incidence from 1975 in 5-year increments to 2008. And what you'll notice is that there's this very clear trend of how age-specific incidence has changed over time in colon cancer. And when we have this stochastic process, we can actually write down the Kolmogorov forward equations for this whole process. And by solving those equations for the probability generating function, I'm not going to go into all the math, but you use the method of characteristics. It's a very cool math problem. You can actually write down what the hazard function is for this process. And so mathematically, it's this limit of cancer development in some time between t and t plus delta t, given that you haven't gotten cancer yet. So this random variable is TC is cancer development. And so when you write down that equation, you can actually fit this model to epi data and pull out some of these important parameters about growth of adenomas and mutation rates. The cool thing also is that with this connection to this model, you can see that the incidence curve actually relates to certain parts of the carcinogenesis process. So in later ages, there's this linear phase of the hazard function that relates to actually the incidence of the adenomas. So it's sort of a backwards process, and the survival of those adenomas with the slope parameter. This intersects with the age axis at some time that's described by the initiation of the adenoma plus the clinical window till detection, that whole time period. And then at middle ages, there's this exponential growth process that's actually described by the growth of the adenomas, so the rate of growth of birth minus death of the adenomas. And then at young ages is this power law behavior that's kind of goes with this classic Armitage-Dawes type multi-stage carcinogenesis process. So you can actually see with the shape of the incidence curve how there's differences that describe the cancer process on average in the population. So once we have these parameters, we can use the model to make predictions. So this is a main project that I did in my PhD, where we looked at this type of modeling for multiple GI cancers using the SEER registry, so gastric, pancreatic, colorectal, and esophageal adenocarcinoma. And again, the goal was to estimate these biological parameters and compare them between cancers using cancer incidence curves. And one thing you'll notice is for EAC, there's quite a rapid increase in the number of cases in this cancer type, which is why we studied it during my PhD. And there's a slightly different shape of the incidence curve. And one of the reasons for that is that this model actually isn't perfect for this type of cancer. It actually needs this initial stage, which we model as a Poisson process of development of Barrett's esophagus with some rate nu of teeth. So I'll tell you about, yeah.
Could I just ask you for esophageal cancer, what is on the left and what is on the right?
Sorry, so the left-hand side is the raw crude incidence for these years. And then you can use an age period cohort modeling for adjusting for those calendar year trends to get the underlying sort of age effect. So I won't go into details of that today, but you can use these type of classic epidemiological approaches to adjust for calendar year trends and then get this kind of adjusted hazard rate. So this is actually what we fit to, and then you can add on these calendar year effects that we think are due to things like changes in maybe screening strategies or guidelines. There's going to be birth cohort effects and period effects that affect incidence. So,
and it seems that in EAC, these is this the birth year effects are really huge, right?
Yes,
Is it
yes.
increasing
So
over time in incidence or?
yes, and especially for colon cancer too, you see that these like calendar year effects have increased, or birth cohort effects have increased a lot in later birth cohorts, which explains some of the early onset cancers that we see. So it's interesting to look at particular at certain calendar years and birth cohorts. And actually the results I'll show you for Barrett's are a birth cohort specific model. So actually there's growth rates that are specific to different birth cohorts and they vary quite a lot. So I won't
Oh,
dive
wow,
totally into this, but yeah, in our papers, you can see about the birth cohort differences.
great.
Thank you for pointing that out. Um, and yeah, so I've been talking a little about Barrett's, but what is Barrett's? So Barrett's is this change in the distal end of the esophagus from normal squamous epithelium to this metaplastic type tissue that looks more like intestine. And the reason this happens is often because of gastroesophageal reflux disease or acid reflux in the distal end of the esophagus coming up from the stomach. And it's an adaptation of the tissue to deal with this acid. And that's why patients don't typically know they have Barrett's, because they are treating their acid symptoms, but really their tissue has changed fundamentally. And unfortunately, for those patients with Barrett's, they're also at higher risk of developing precancerous lesions of low-grade and high-grade dysplasia and eventual cancers. And so finding patients with Barrett's is very important because they're at this high risk of developing cancer. And so one question we asked,
Sorry, again,
yes,
this is
yes,
very interesting, but I have another question because
yes,
we have been trying to understand this in our group as well. Do you understand correctly that the normal esophagus does not contain crypts and then the Barrett's does? Or like that's a little
yes.
different from the colon, right?
Yes, yes. So the normal esophageal lining is this stratified squamous epithelium that's more kind of like the skin. So there's no crypt like, and so actually esophageal squamous cell carcinoma has a different evolutionary trajectory because it grows more like a stratified epithelium type of cancer, like skin cancer. But for esophageal adenocarcinoma, it has to develop from this Barrett's, which we'll talk about, which is this crypt-like structure. So different than normal colon because this is a stratified epithelium normally.
Great, thanks.
Sure. So when we were talking with the clinicians about some of the knowns and unknowns in Barrett's, I'll run down some facts for you. So again, like I said, Barrett's is typically asymptomatic. People are treating the acid reflux with things like proton pump inhibitors, but they don't sort of know they have Barrett's. The prevalence is about 1 to 2% in the population we know from sort of population studies. And these patients with GERD have like a 5-fold higher relative risk of developing Barrett's. For this reason, these high-risk patients with GERD are often suggested to get screening at age 50, which that number will come up again in current guidelines. We also talked about how the rate of progression to cancer in non-dysplastic Barrett's is very low. So to go from non-dysplastic, which looks like this kind of salmon-colored mucosa, to these highly fatal cancers is very low. And we know that there's about 10,000 cases of EAC in the US per year. However, determining who with Barrett's will later be the ones to progress to EAC and who will remain cancer-free is this large clinical challenge of risk stratification. And it was also unknown whether all EAC actually comes from the Barrett's lesion. And the reason that is sometimes people come in with these large cancers and you don't necessarily see the nice Barrett's around it. So it was kind of unknown whether this was like an obligate precursor to the eventual cancers. So we tried to answer this question using our modeling. And so this is a nice segue into talking about birth cohort modeling. So we use those cancer incidence curves from SEER in a birth cohort specific way for men and women separately, because you can see that the incidence in men is much higher for EAC than it is in women. So they need different rates of progression. And with this birth cohort specific model, we can then answer some questions. So we have our model fits to the data, and then we can say what's outputted from the model. Since we didn't use any of this information as input, we can see what the model says it should be. And it says that about 1.6% of the US population has BE over age 40, which is about what we think it should be. It is from the model that the annual progression rate is about 0.1 to 0.2% in men, which is around what we think it is. And then when you add up the total number of cases using something like a life table for the person years at risk at some age and some birth cohort, multiplied by that hazard function at some age and birth cohort using our model parameters, you look at the cumulative incidence of that, and it's about 10,000 cases. And so all of this adds up to say that all of those EACs that we see in the registry must have come from this Barrett's if we believe all these clinical progression rates. And there's no cases that are sort of missing that would have come from a non-Barrett's pathway. So this was quite interesting for our clinical collaborators because it says that really Barrett's is the population that we should be focusing on because they contribute to nearly all of the cases that we see in the population. And so the next question we could tackle with our modeling is then what age should GERD patients be screened for Barrett's? And with this, we can think about this problem again in terms of kind of a quantitative approach where we're looking for these windows of opportunity where someone has developed Barrett's, but they have not developed cancer yet. And so with our model, we can answer more than just a hazard function. We can look at things like probabilities that some precursor has developed and that the fact that some symptomatic cancer detection hasn't happened yet too. And so with the same type of model, we can write down these probabilities sometimes actually analytically and try to solve for these sort of windows of opportunity of what is the optimal TS where you're going to catch the most Barrett's patients and have the highest yield. And so when we write down the probability for what we want for Barrett's screening was what screening age do we want to be after this random variable of Barrett's onset, before this random variable of cancer onset, and also importantly, that this time of screening doesn't happen too late in life that there's a high probability that you would have died of other causes. So you can use sort of mortality tables to also condition on this. And we call this sort of an objective function that we're trying to maximize. And so again, when we look at what the model predicts for Barrett's prevalence in both the general population and the GERD specific population, we can write down this equation for the cumulative probability or the prevalence of Barrett's in the population, given that these patients don't have cancer yet and they're still alive, and compare that to what the clinical data shows. The clinical data is in the dotted lines and the model predictions are in the solid lines. And this was actually the clinical data that decided that 50-year screening age by looking at, looks like the prevalence is sort of teetering off after 50. So again, not very much data for the screening age of 50, but what we can do with the model is not necessarily look at this conditional information on patients that we know don't have cancer and we know haven't died yet, but look again at this joint probability of when is the best time to screen if we knew the progression and the mortality rates of this population, and then find those optimal values. And so we published this a few years ago, looking at both men with GERD and women with GERD, and saw that actually probably the optimal time is a little later than 50 to get the most bang for your buck. And the way to look at that from also sort of a public health standpoint is, well, what are your costs and benefits? And you can quantify this also by looking at sort of what is the number of endoscopies that you sort of save if you screen later versus how much cancer do you add at risk. And we saw the cancer risk is still very low if you screen later, and you'll save like $20 million per book card or something based on the number of endoscopies that you'll avoid. The cool thing about this is you can compare this to trial data. So in the UK, there was a very large trial called BEST3 of screening for Barrett's. And they saw that the prevalence of Barrett's was around here at this age group. So pretty in line with what we saw as well. And what was published last year in Gastro is they took just that trial data and had some sort of statistical model for explaining progression, and actually found really similar optimal ages when they took this totally different approach of just analyzing the trial data. And with all of that together, they were able to design this new trial that just started in the UK for 120,000 patients with GERD. And they decided to initially screen the men at 55 and the women at 65. And so this is why I think the math models actually can be really helpful, because it helps to determine these very large trial designs so that you know the most efficient strategy before you start spending a lot of money on a trial. So that was a nice verification of what we found as well. In this way, you can kind of iteratively determine the next optimal surveillance window with these conditional probabilities. And so we answered some other questions in this paper, like, should you rescreen after a negative screen for Barrett's? And you can do that with conditional probabilities. Okay, so now some newer stuff in review. So how do BE genetics play a role? So a cohort that we study a lot in my lab and with current funding as well is a large case control study that was done in Seattle, Washington at the Fred Hutch Cancer Center, where they looked at patients with Barrett's who did not progress to cancer since the start of the study, which they call controls or non-cancer outcomes, versus patients who do progress to cancer, which they call cancer outcomes, who are cases. And what they did was take biopsies and actually isolated the cryptoepithelium and performed whole genome sequencing of those biopsies to compare certain genetic features in those cases versus controls at that initial time point before there was development of cancer or non-progression. So they found various things. So they found that cancer outcome patients have more TP53 mutations. So that was sort of expected as an initial driver event. And you can see in the first line here, the cancer outcomes have, a lot more TP53 mutations than the non-cancer outcomes. They found more copy number alterations and more structural variants in those that progress later to cancer somewhat going up to the levels that you see in actual cancers. And they also see more extrachromosal DNA structures, which are these circular DNA structures. Some other things that we've looked at are, do you think that there's faster growth dynamics and increased biological aging? We've published a bit on this in the past and are still working this space, but I'm not going to talk about these projects today. What I want to focus on today is actually the microbiome that we've analyzed from this data. So why do we look at the human microbiome? This has kind of been a new space for us the past 5 years. And one of the reasons is that there's really so many bacteria in your body. So there's about 1-to-1 bacteria to human cells in the human body. There's thousands of unique bacterial species that reside in our bodies. And we know that some microbes actually cause cancers. So things like human papillomavirus causing cervical cancer and some type of oral cancers. And then, as I mentioned, Helicobacter pylori is a cause of stomach cancer. Other microbes might modulate carcinogenesis in some way. So we know that PTS positive E. coli can cause a certain mutational signature in colon cancers. But most of these microbial species are totally commensal. So they've evolved with us and they actually help our bodies in many ways. So there's these very interesting studies coming out trying to disentangle the role of these microbes in cancer. And we know from lots of interesting work coming out that they modulate things like growth and immune evasion. And so super interesting things. But we asked sort of a different question. So once these bacteria colonize certain organs, they replicate and die and have their own population dynamics happening within their communities. And so we wanted to know, how do these communities of microbial species evolve in the human body in sort of an ecological sense? And so what my PhD student, Caitlin, initially did when she started her PhD was look around the literature and see what has been known about microbial diversity at these various stages in Barrett's through cancer progression. And at kind of a phyla level, there wasn't too much differences across the studies in terms of changes in phyla, relative abundance in, say, normal Barrett's, hygrodysplasia, and esophageal adenocarcinoma. But they would pick out sort of specific species and say, oh, the relative abundance between Barrett's and cancer is different. Maybe that's an important microbe. But we kind of wanted to ask, well, how did all these communities get here to begin with? Can that be described by some sort of population dynamics? And so to do that, we set up this model that was based on some nice ecological modeling work in microbiome field previously. And what we looked at was sort of the esophageal microbiome as the space of communities where the different taxon or species can have some sort of birth rate and they can divide and they can also die. And also to kind of keep these dynamics from just coming to saturation with whoever is the highest prevalent microbe, we also allow for immigration of microbes to come into the esophagus, stay through the mouth, or maybe the proximal stomach because of gastroesophageal reflux disease, or in Barrett's, maybe it's just from the normal squamous epithelium. So you can think of different scenarios for this model. The simplest, kind of most boring, I guess, is the neutral case where all the microbes have the same birth rate and the same death rate. So they all have the same growth happening in the community, or a more general non-neutral model where you can have taxa-specific birth and death rates of the microbes. So some nice math has been done over the years, and we followed some work from Arne Charleson's lab that looked at the transition probabilities for this stochastic process. And when you look at in the large population size limit, you can write down a Fokker-Planck equation for these transition probabilities. And at equilibrium, you can actually solve for the stochastic process for this Fokker-Planck equation. And then you can fit to data. So then you have an equation for occurrence, which is what we get from taxonomy tables, and equation for mean abundance and this migration rate parameter. So again, won't get into the details, but you can check out Caitlin's preprint on this and some amazing work from our colleagues here. So what does this look like? So in the neutral case, you can apply this to certain data sets, and what you get is an expectation is sort of this S-shaped curve. And the general kind of intuition is that the more abundant you see a certain microbe in a population of samples, the more often you would expect it to occur across the samples. And then you can write down sort of statistics for how well does the data fit that neutral expectation, and write down some R-squared statistics for the distance from that neutral expectation. So previous groups in Arne's team, they looked at compost and different bacteria in compost and saw that there's a fairly good fit to neutral expectation of the community assembly. They also looked at wild populations of mice and can quantify certain taxa that seem to be very far from this neutral expectation, and maybe there's something different going on with the growth and selection of maybe pathogenic types of bacteria. And it's also been looked at in human populations. For example, in this study, from China looking at rural versus urban cheek swabs and trying to determine if there's effects of kind of more urban population in terms of the assembly of certain microbes. But none of this has been applied to cancer and not to precancer, so we tried to apply it in our scenario. And so we accumulated whole genome sequencing data sets from the literature from normal squamous epithelium, from gastroesophageal reflux disease, histological confirmed biopsies, from Barrett's esophagus from the study that I mentioned, and also from esophageal adenocarcinomas as part of ICGC, and a small validation set of EACs that were published with whole genome sequencing data. The pipeline that Caitlin developed for analyzing these samples was take the whole genome sequencing, remove all the human reads from that computational, from that whole genome sequencing data set, which we'll talk about in a second, mapping those reads potentially to this microbial database that has no human reads in it, removing any microbial hits that don't seem to have very good genomic coverage, which I won't talk about this tool today, but really important for being very stringent about making sure that these are real reads that map to taxa, and then doing the bioinformatics. So the computational removal of host DNA we published earlier this year, and it was really interesting because Caitlin looked at what was it, about 5,000 metastatic tumors in the Hartwood Medical Foundation. And when she only did alignment to HG38 for removal of the human reads, when you look at the diversity of the microbiome in those samples, you saw this very clear distinction between men and women, which is not expected in terms of general microbiome space. There shouldn't be that much distinction between sexes. But then when she added in the newest build of T2T, she saw that that sort of separation was removed. And the reason for that was a lot of these reads were being missed that were now more accurately mapped to the Y chromosome, because there's more of the Y chromosome that's part of T2T. And they were erroneously getting through the pipeline and going through microbiome analysis. So really important to remove human reads accurately, and so important that we looked at fecal samples, over 300 fecal samples from a paper studied in Nature Microbiology a couple of years ago, and saw that if you don't do this proper host filtering, you can actually, from just metagenomic sequencing of stool, actually positively re-identify participants, which is very interesting because usually people publish these data sets and they think that it doesn't have human reads because it's mostly bacteria, but actually there's enough human reads in stool to actually do this. So once she applied these more stringent pipelines, that ability to identify patients from their stool was removed. So important in terms of kind of patient privacy and how we publish data going forward as well. Okay, so what about our data sets? So when we looked at whether the neutral model was appropriate for some of this data, we saw that in normal and GERD samples, the neutral expectation fit fairly well. So R-squared is over 85 for these data sets. And in Barrett's, we also saw fairly high fits to the neutral model with some exceptions. And the one that I'll point out just for nature of time for today is the non-cancer outcomes or the non-progressors of Barrett's. Their position of Helicobacter pylori in this occurrence abundance plot is pretty far from the neutral expectation. And we'll talk about that in a second, but you don't see that same thing happening in the people who eventually progress to cancer. When we looked at EAC, everything seemed to be changed. So you see very poor fits to the neutral model, a lot of taxa looking much different from this neutral case, and seeing that also in this much smaller data set of EAC samples. I'm not going to talk too much about the EAC scenario, but I will focus on the Barrett's for one example for Caitlin's work. And why we were interested in H. pylori is that although HP causes stomach cancer, we know that it's associated with reduced risk of EAC in patients, and it's not really understood why that is, and if that's totally true, but there is some evidence for that. And so to look at this closely, we said, okay, well, what if we look at a more general model and not just the neutral model in this case, and we actually change some of the parameters for H. pylori in particular and see if we get a better fit to the data. And so for that, we have to move to simulation. So we used a Gillespie algorithm to simulate this stochastic process where we assume the same birth and death rates for the other taxa and some different growth rates for the H. pylori taxa. And so we see when you assume total neutrality, you don't get very good fits, but when you give H. pylori a growth advantage, you start to see data that looks more like the empirical data. And we went through many simulations of different scenarios and different parameter types. I won't go through them all, but cutting it to the chase where the kind of necessary thing that you needed to have close fits to empirical data was this higher growth rate. So either decreased death rate or increased birth rate for H. pylori versus all the other microbes in the population.
Sorry, Kit. This is very interesting. I'm trying to understand. So basically, on the left, you're showing that H. pylori has kind of a higher abundance than expected, based on how often it occurs in samples. And then when you look at the two plots on the right, why is the one on the far right fitting the data better compared to the other one?
Yeah, I rushed. So this one was assuming that all taxa have the same growth parameters. So the totally neutral case, which, somewhat looks like the data, but not perfectly. And then we know that HP was not in a place that it looks like on the empirical data. So we started to play with different growth rates for HP in particular. And we allowed a little bit of noise for the other ones. That's why there's a little bit more noise of where they occur on the occurrence abundance. But in general, you need this scenario of having either a lower death rate or higher birth rate or both for HP to see it kind of move in that direction where there's higher abundance, lower occurrence frequency, which when it's looked at in the past, I mean, no one's quantified it in the esophagus in the same way that we have, but they always look at sort of the higher mean abundance, but they don't think of this scenario where it's actually lower frequency as well. And sort of what can explain that type of occurrence abundance in the population? And so we're quite interested in this, and it needs much more mechanistic experimental work to sort of prove any hypotheses. But one hypothesis might be that these patients are actually responding to their acid reflux medication better, because we know if there's lower gastric acid, H. pylori grows faster. And so even though they're all on PPIs, there might be some sort of, something about the non-progressors that they're actually being able to control the acid better. And again, getting this type of data, clinical data on specific pH measurements over time takes a lot of planning, which was not part of the clinical care in the study, but it was more hypothesis generating than anything.
Thanks.
Yeah. And I'll just mention for the EACs, in order to get that kind of crazy looking plot that we saw, you need very different growth rates across many taxa. So we think that, there's a lot more niche-based processes, ecologically speaking, in cancer than there is in precancer and normal tissue. And that's sort of the gist of the paper. Okay. So moving on to IBD for the final part of the talk. So ulcerative colitis is one of the two main subtypes of IBD that we study in the lab, and the incidence is about 1 in 200 people. And what happens for these patients with colitis is that they have some uncomfortable chronic conditions like diarrhea and pain and bleeding. They're often treated with immunomodulators, biologics or 5-ASAs. And what happens is over time, they have these bouts of inflammation that cause these ulcers to form in their colon tissue that can be diagnosed with colonoscopy. And on histology, you see a lot of infiltration of immune cells. And so it's an autoimmune disease that probably has genetic predisposition, which I'll talk a little bit about, probably some influence of the gut microbiota for sure. And also, of course, these immune-driven responses and differences between patients. So many people in the call, including Ivana, have studied sporadic colorectal cancer a lot. And we know that there's these specific kind of genetic events that often occur, like loss of APC mutation and late p53 mutations in colon cancer progression. And in colitis-associated colorectal cancer, it's kind of the opposite. So you have early p53 mutations in this setting of chronic inflammation. You develop these low-grade lesions similarly, but sometimes they're more flat in shape. And then loss of APC is often a later event. And unfortunately for people with colitis, this progression happens much more often than sporadic colon cancer. So there's about a 2 to 4-fold higher risk of CRC in the IBD population. And so those patients are suggested to undergo lifetime surveillance for this reason. And so surveillance, we know, has good outcomes. So there's earlier cancer stage at diagnosis and improved survival. And the 10-year risk of colon cancer when low-grade dysplasia is diagnosed is about 30%. But the challenge is, again, knowing who with those low-grade lesions will progress to cancer and who won't. And we had a paper a few years ago kind of describing those kind of genetic changes that happen at those various stages. But there's also morphological differences. So these are all low-grade lesions in patients with colitis. Some are these more polypoid shapes, which is sort of a more classic polyp shape that are easier to remove. And some of these more flat shape called non-polypoid lesions that are a little more difficult to remove. And so part of my postdoc was actually motivated just from the clinical need of describing risk to patients so that they can make some important decisions. So a lot of patients with IBD are diagnosed with these lesions in their 30s, and they're making decisions whether they should watch and wait on surveillance for the rest of their life, or if they should undergo some sort of invasive prophylactic surgery like partial colectomy of the colon. And so the first thing we did was just look at the clinical variables and create sort of a statistical model for cancer or high-grade dysplasia progression in these patients with low grade. And with a tool with just 4 clinical pathological variables, we were able to validate the prediction and risk stratification of a population with low-grade dysplasia in terms of many years of follow-up in the UK cohorts that we looked at. And just for a little bit of math, it's pretty simple. So when you have a Cox proportional hazard model, you get out things like the beta coefficients of the Cox proportional hazard model from hazard ratios, you get some baseline hazard, and then you have the patient-specific variables, and this is how you can write down an equation for risk into the future given some time with that model fit. And what we did was build this into the back of a tool that doctors can use where they can input with the patient in front of them those variables of things like, is the low grade large? Does it have severe inflammation around it? Is it multifocal? Was it unable to be totally resected and some tissue was left? And then show patients kind of their predicted risk over time. So this is my colleague Misha in the NHS hospital using the tool in the clinic. And it can also have these kind of charts that are useful for patients to understand their risk, where they may be able to kind of digest the information better than percentages by saying, okay, 100 patients like you with a similar risk, we expect this number of patients in the first year to progress to some advanced neoplasia, this number of patients within the next 5 years, and this number of patients in the next 10 years to help them try to make these important decisions with their doctors. And so a lot of the work that we do now is how can math models sort of help this kind of more statistical based approach. And so a big project that we've been working on the past several years was funded from the Veterans Affairs. And we call this project Mosaic, or Mathematical Optimization of Surveillance Ages to Intercept Colitis-Associated Colorectal Cancer. And we have access to both the whole clinical database of veterans in the US, which is a very large population of a generalized healthcare system, and also something called the Million Veteran Program, which, if you know, the UK Biobank is quite similar setup. So they have volunteers, veterans who've donated blood that has been arrayed for germline analysis. And a lot of studies are funded through this program to look at things like disease associations. And so we were interested in colon cancer in this population. And just for some numbers, in the whole VA, there's about 80,000 patients, we think, with IBD colitis, and about 5,000 of them have developed cancer. In MVP, there's about 12,000 with colitis and about 1,000 cancer cases. And so the goal was to determine germline variants associated with polygenic risk of colitis and CA-CRC, and also to use some modeling to help with this surveillance problem. So is it the best idea to start screening at 8 years, for example? What should these screening intervals be? Can we do better personalization of these screening intervals? And so this allowed us to delve into a kind of new area in the lab, thanks to Brian Johnson, who's a really talented PhD student with me, where we had all these clinical data trapped in these SQL tables. And what Brian did was build these large language models to extract useful information from this longitudinal EHR data that we have. So we have data on 14 million patients from veterans from 1999 to 2022. And looking at pathology reports, these models can pull out things like cancer diagnoses, things like duration of disease, and a lot of other variables. And so, for example, a lot of times people will look in EHR and look for ICD codes for diagnosis dates of certain disease diagnoses. And what we found is that actually the initial ICD code is often not a great proxy for the actual diagnosis date of a disease that was maybe mentioned in some other clinical note that's more accurate. And so, for example, you can give some prompt to an LLM saying, look at these notes, determine the length of time from this note in the patient's original diagnosis, and feed it a bunch of clinical notes, and it might be able to tell you the answer. And we noticed that this is actually more accurate than using something like an ICD code. I know I'm brushing over a lot of biomedical informatics nomenclature, but basically pulling out things from notes is a hard clinical problem with these large databases that we're trying to solve. For this reason, we had a paper recently accepted doing this in the VA cohort, where we use these LLMs to pull out things like colon cancer diagnoses, high-grade dysplasia diagnoses, low-grade dysplasia diagnoses. And these open weight LLMs can be uploaded into the system and read millions of notes. You have to use a GPU if you're doing some of these hard problems, but we were able to access one in the VA. And there was just no human-led development. We just used some of these LLMs off the shelf for these tasks. And we saw really high accuracy in pulling out these diagnoses from things like pathology reports. And And this has been pretty amazing because with the use of these tools, we're able to kind of use these large scale databases that have been previously unusable, basically, for some of these big downstream diagnoses. And I think this is such a cool area for mathematical biologists that are interested in combining clinical data, because we can actually have these really big structured data sets. And so, for example, this is just some preliminary results trying to validate that UC care tool in the data. And now we have something 5 to 10 times as many patients with low-grade dysplasia than we had in the UK. And we can actually validate that these risk stratification still works in the veteran population up to 25 years of follow-up. So really exciting clinical work, but also data for mathematical biologists opening up. And so going back to our modeling, we wanted to use a multiscale model for understanding cancer risk in these patients. And for this, we used the LLM to get the cancer cases, age-specific cancer cases diagnosed in the VA, and fit these hazard functions that are now specific to colitis-associated colorectal cancer. To do this, you can use a likelihood-based approach based on the observed cases that you expect, and fit the model that way. And just to write down again some equations, actually, this model that I was describing can actually be approximated very well with a simpler model. We have an effective mutation rate, a malignant transformation, and then a time lag that corresponds to the age of the tumor. And you can write down an equation and fit to the data. When you do this with just this cancer data, the fits are not that strong. So you don't have perfect convergence, say, of an MCMC on these estimates because you just don't have enough data. So how do we add more data to make this a better fit and be able to actually pull out all these parameters, or at least most of them? And so we asked, what other data can we use? And so some really preliminary results I'll show you now is how to dive into actually the pre-cancer incidence data. So looking at just this part of the stochastic process and trying to jointly fit with the cancer incidence process is what Brian's been working on now. And maybe you can use genetic data, which I'll mention briefly. And so the pre-cancer incidence data has been really interesting. So when you looked at just the initial colonoscopy of these patients, you can also look at the probability that they'll be diagnosed with some low-grade lesion by age. And to fit this to the model, there's actually already been some nice math work published that writes down this process for a Poisson initiation of a low-grade lesion that then undergoes a birth-death process. And I have math in the backsides if you want it, but basically it's this integral where you have some probability of detecting the lesion of a certain size at some time, multiplied by the probability that they have not developed cancer yet, which is important in this data to condition on to get accurate fits. So now we have an equation for the hazard function, we have an equation for the adenoma prevalence, and we can jointly fit them, which is what Brian has recently done. And so the data is shown again in the kind of dots and the error bars, and then the model fits are shown in the lines. And so when you do this, you get fairly good fits to the data. So these are paired plots for things like the mutation rates of the initiation events, the growth of the low-grade lesions, the effective mutation rate for malignant transformation, and this time lag of clinical symptomatic detection. There's still quite a bit of correlation between the parameters, even though they do converge nicely. So we're talking about more data to add to this to fit to, and there's been some cool math recently published about adding in things like tumor size at the end that we can also fit to in this data, because we can pull that out from the pathology reports. But pretty exciting to use data that we've never had before. So things like adenoma prevalence by age, which we can determine from these large scale databases. So comparing to some previous work, now this is older work that I showed you at the beginning, was published in 2013, but we can look at some of these evolutionary parameters from the sporadic case and compare them to the IBD CRC case and see what sort of parameters are different and which are similar. And so one thing is that this lambda, which is the slope of the hazard function, which is corresponding to the initiation of those low-grade lesions, is much higher in the sporadic population, which we actually know is true. There's a higher prevalence of adenomas in the general population. There's about 40% of us have a low-grade lesion at any time that we don't know about. And then lower low-grade risk in IBD. Similar lag times or the sojourn times of the tumors, but much higher malignant transformation rate in IBD. And that's sort of an interesting hypothesis that they're making malignancies quite frequently. And then the time to progress to clinical diagnosis is actually quite similar. So you can start to compare these things. We need to refit this with more recent data, but I just wanted to show you what you can do. And of course, all of this is sort of looking at the mean population situation, right? And we think that's important for screening, because if you don't know anything about the population, you better make some decisions based on sort of the average time to progression, et cetera. But how do you personalize this? And I think this is like a really interesting area. And we did a little work of this published a couple of years ago in melanoma, where we fit these multi-stage models to melanoma incidence. And then we had sort of an average tumor age, or the sojourn time, TLAG, in the population. And then we also had whole genome sequencing of the tumor, so we could look at things like age-related mutational signatures as another proxy of aging. And combined with the average age, you can use kind of a linear model to adjust the average TLAG for that patient-specific tumor mutational burden. And so then you have like a patient-specific tumor age. And then you can look at things like in this paper, we looked at polygenic risk scores, what those onset ages were in certain quintiles, and also what they were in terms of certain autoimmune alleles that we had information on because we had whole genome sequencing. And I just love this example of trying to combine kind of epidemiological data that we know these parameters make sense in terms of the clinical outcomes, and then also trying to use omics to personalize them. We also had a paper a couple of years ago published trying to measure these growth rates with single cell data. So we had some work in blood cancer where we looked at the phylogenetic trees of single cell derived clones. And what Brian did was look at these phylogenetic trees with some mathematics collaborators here at UCSD. And we wanted to know the growth rates of these subpopulations or these driver clones in myeloproliferative neoplasms and in normal individuals by looking at the shape of these trees. And I'm not going to dive totally into all the math to have you look it up, but just as an example of how to derive this from more types of omics data, we were actually able to write down the equation for what the growth rate should be based on the lengths of these internal and external branches of these phylogenetic trees, and then quantify in a patient-specific way what the growth rates of these driver clones are. So this is other type of data that you might be able to combine with epi data and personalize these growth rate estimates. And just to highlight why this was so important is when you divided the population in these Nature papers that we applied the mathematical theory to into sort of the higher growth rates and the lower growth rates, those with the higher growth rates, which is this initial exponential growth phase right after the cancer is initiated, they progress to diagnosis much faster than those that had the lower growth rates, which might be sort of intuitive, but interesting to really quantify. And also when we had information on two clones, so for example, this patient, some studies also published bulk sequencing data over time of those individuals. And we were able to quantify the size of those clones over time and see that actually just with single time point estimates, looking at the phylogenetic tree, anecdotally, we're able to predict ones that will outgrow and ones that will decrease in size based on single cell data. And just to highlight the importance of these evolutionary parameters, if you were just to look at something like the number of cells with that mutation, just the absolute fraction of the population that has that mutation, that does not split the population in terms of who progresses fastest. And I think this is where like mathematical biology comes into play, where it might be natural to think of, oh, maybe the biggest clone, but that actually at this time point already tells you that the clone was big enough to be detectable, and nothing about the real kind of risk inherent in that clone, which is where math can actually derive these important parameters out. Okay, I think I'm running a little low on time, so I'm going to rush forward and maybe skip this section talking about specific alterations in IBD, because I want to leave some time for questions. But basically, we've done a lot of work of whole genome sequencing of samples of IBD over time and trying to look at clonal changes and growth within the population to, again, try to use genomic data to sort of personalize some of these estimates and get better targets. Okay, the last slide I want to show you is about germline work that we've been doing. So with Million Veteran Program, we have all of the SNP data, and what Hiram Eddington has been doing in my group is looking at new ways to derive polygenic risk scores for cancer and colitis in these populations using machine learning based approaches. So he used XGBoost to combine SNPs that we know are involved with colitis associated risk, and also importantly, HLA alleles that we know are involved with certain types of colitis disease severity, for example. And what he was able to do is kind of do a PCA on the Shapley values of these and light and cluster them and see that there's actually genetic subtypes of IBD, which is a really interesting way to look at this. And we were interested in if this is, associated with that much disease severity or anything. And when you kind of break it down, you can look at specific clusters, for example, 1 and 6, and see that there's different importance of these Shapley values for different areas of the genome. So example, for cluster 6, there's importance of certain HLA alleles that has been published previously for things like response to therapy. And then cluster 1 might have some other sort of like IL-23R type of autoimmune related diseases. And so when you combine this with other clinical variables and do a Cox model for something like cancer risk, we see that actually these clusters hold weight. And so this is sort of an exciting piece of kind of personalized medicine where there's this kind of underlying genetic architecture that we're not taking into account when we do just linear polygenic risk scores that kind of come out when you do this machine-based learning approach. And when you do this, you can actually risk stratify for many years out into the future with just some basic clinical variables in this genetic data, which we're looking to validate in some other biobanks now, but kind of exciting work trying to combine germline genetics and other kind of statistical genetics approaches. Okay, so in summary, evolution from normal to cancer cells requires time. These surveillance exams is what we've been utilizing to measure things like field cancerization. Mathematical models can combine epi and population dynamics to infer things like growth and mutation rates. We've been looking at host cell populations, but also microbial cell populations. We've been applying LLMs and EHRs to get summary statistics out from an epi level, but also molecular data can serve as inputs and targets for math models. And although translational bioinformatics helps share decision makings, I didn't talk about this copy number alteration biomarker that we published this year in IBD. These reliable mathematical models are needed to predict these optimal strategies. So biomarkers are only going to go so far by themselves. You need a model to actually predict what are the outcomes in patients. Okay, with that, I think I'm just going to wrap up. Thank my lab here at UCSD. Couldn't do any of this work without them. Other collaborators also at the San Diego VA, and showed some previous work from other collaborators and all of our generous funding. So thanks again for having me. Happy to take some questions.
Thank you so much for the wonderful talk. Please post any questions you have in the chat. And while you're doing that, I'm going to ask some questions. So I'm very interested in the model for you had where you are trying to fit adenoma and cancer incidence data. And basically, my question is about parameter identifiability. What are the limitations there? Do you think adding more data will help, or like what is the current state and what is the best you can hope for, kind of?
Yeah, thanks for this. There's lots of cool research done in this space on identifiability. Some from Marissa Eisenberg's group has some of the best papers on this, but basically with like the MCMC fits, you can see a lot of the practical identifiability. And so we know that if we have too many parameters, the estimates don't work and you won't see any convergence of the MCMC, for example. We see better fits with more data we have, if it's useful data. And again, mathematically, you can actually determine either practical identifiability versus sort of structural identifiability. And of course, we're really interested in practical identifiability because we want to actually know these estimates from data. And so I'll confirm that the more data you add, if it is useful data, will help you to infer more parameters. And so from those kind of plots, you can see and traces of the MCMC, whether you're getting convergence and if you've got multiple maximum by looking at the trace plots. We also do maximum likelihood estimation. You can see like how big those error bars are. So there's different approaches, but we have to fix some parameters. So like I showed you, there's about 4 parameters that we can estimate right now. And we have to fix some of the other ones, which we've done in previous papers, did some sensitivity analysis based on how sensitive the fits are to those choices. And so I don't have time to dive into it, but it's a really important question. And you'll see a mess if it's going to be not identifiable, but there are some intricacies that you have to be very careful with.
And this is kind of connected to the question from the chat from Rui Bojang. Is parameter identifiability an issue when using multi-type branching processes, say for Barrett's esophagus, to fit the incidence? How would you validate that the model has a correct number of stages?
Oh, that's a really good question. So the number of stages can be handled in different ways. We've in the past, for the Barrett's model in particular, we did a likelihood-based approach. So we added more stages and then did like a likelihood ratio test, whether you get any improved fits, trying to find sort of the most parsimonious models. You can add more mutations, you can add more growth stages, but at some point, yeah, your data is not going to be able to fit that many. So we try to go for sort of a parsimonious approach. There's also cool approaches now using kind of simulation of different trajectories and trying to infer sort of with like a neural network, which ones were the most likely. Again, Marissa gave a great talk about this at SMB. And so you want it based in biology, but also you don't want to overfit the data. And I think there's various approaches, but likelihood ratio test is one way to do that.
And I have one more question. I would like to understand the polygenic risk scores. I have
Yeah.
never seen them used in the setting of cancer risk
Yeah.
like this. So if you can go to maybe one of the last slides you showed.
Yeah.
So this is just germline.
Yes, this is just germline. And so in MVP, they've got the genotyping data, which you can do classic GWAS, which is what this is. And this is one that we did. And you can see that IBD has these really strong hits in certain genes. Some of these are in HLA regions already. And some of them are other kind of like autoimmune related genes. This matches up like almost perfectly with what was found in UK Biobank. So this is like a really strong IBD signal. So we pull out the SNPs that are significant there, but also nicely in MVP, they've already done the HLA typing. So you have all the HLA types of those. So we didn't have to do that ourselves. But we just collated this list of like things in the literature that have been associated with IBD and then put them into this machine learning based model to pull out the coefficients of and allowing for this non-linearity rather than just a linear based polygenic risk score. There are linear based approaches for sporadic colorectal cancer out there. None of them have really been, I think, used clinically, but when combined with clinical variables, they do give you some boost in sort of like AUC of cancer risk. But we wanted to sort of dive deeper. And because there's lower numbers of colitis-associated colorectal cancer, if you just do sort of a GWAS of colitis-associated colorectal cancer, we're not getting significant hits. But we didn't let that stop us because we think we're pretty underpowered compared to sporadic. And so this was like totally driven by Hiram, and Hannah Carter is the co-advisor who's been doing this in other disease spaces as well. And so when you take these kind of Shapley values, which is these like important parts of the machine learning coefficients, you can manipulate them in PCA and then clustering to sort of look at these genetic subtypes. But this is a very kind of new way of looking, I guess, at polygenic risk, which I'm excited about because I think there's some underlying biology that you miss if you don't look at it like this.
Great. Yeah. Any other final questions on the chat? Well, if not, I would like to thank you, Kit, again for speaking in the seminar. It's been great to learn about, I've known about some of the work, but it's great to hear about all of the different projects that you are doing in your group. And it's really exciting. So thank you.
Thank
Thank you so
you.
much.
Thank you so much. Have a great day, everyone.
Okay, I have stopped the stream.
Okay.
Yeah, no,